All right, so our next speaker is Joachim Schultze from Bonn. Who'll talk about, I think, more clinically oriented sequencing.
Yeah, so thank you very much, Peter. I'll start here. So I'll bring you a different view a little bit. I'll also try to bring you some of the computation, but since I'm actually trained as a physician, I would like to bring you that view of how we think this is important for the clinics. And that's by purpose. You see that I say from single cells to patients, a lifetime journey. So I really mentioned already this big project that we're trying to establish in the future in my title, and I try to come back to that at the end. This is my motivation for doing this kind of things. You see this is the newest from the WHO about what are the most deadly diseases in the world and in Europe. And you can already see that it doesn't matter where you go in the world. It's ischemic heart disease, heart attack, stroke. Then there's COPD, chronic obstructive pulmonary diseases, which is number 5 here. And you see even in Europe and also in the States, Alzheimer's and dementias are those big killers that are basically problematic. What's interesting is, and that's why I asked Barbara the question, all these diseases that you see here in the top 10 would not work if the immune system wouldn't go wrong. So without the immune system that goes awkward, we would not have these diseases. Yeah? So, and even the discussion even about Alzheimer now is very clear that there is a neuroinflammatory component which is very important. In the field, we're still struggling to know whether this is early or late, but there's no question without the immune system going awkward, we don't have these diseases. And this is even true for ischemic heart disease. There was a very nice paper in Nature Immunology very recently clearly showing that if you have a response that's not going perfect, you will have really massive problems, and that's the macrophage-driven disease. Why I'm saying that? Because everything you see in red is diseases that we are really working on in collaboration, and I will come back to that. And my true passion, actually, from my days where I was trained in immunology, is the myeloid cell component now. And basically, we have them in many flavors. We mentioned them already right now. There's tissue macrophages. There's a lot of granulocytes, monocytes in the blood, and a lot of dendritic cells. They're very important for many aspects in physiology, from ontogeny, homeostasis, to differentiation of whole organs, but then basically they're involved in all kinds of major diseases. So everything I showed you before, and also some more rare diseases for sure. So it's very important to know how these cells are actually in a normal human being under homeostasis, so that we better understand how these are going wrong in the diseases. And that's why we basically worked on this issue for a long time. And we're now already in the second stage, basically. We came up 2 years ago with an idea of how we basically use single-cell technologies to understand what kind of myeloid cells we have. But I would like to bring you to a next level, basically, to a journey to see, like, if I make a map once, that's not sufficient. We probably have to make maps over and over again in an iterative process, and I will share you some of our ideas how to do this. So this is basically my first part of the talk, how we try to build consensus maps for the human myeloid cell space. So this is the world before we started all the single-cell RNA sequencing. So the immunologists were doing that for quite some years. There was also a Nobel Prize about dendritic cells, and we all had the feeling that there's 3 types. We called them with names: CDC1, CDC2, plasmacytoid dendritic cells, because they're close to plasmacytes. And then there's 3 types of monocytes: a classical one, an intermediate one, and a non-classical one. So that was the immunology world. And this world was not always very harmonic, because there was a lot of fighting about whether this is the right thing or the wrong thing. And people did that based on markers, and you see a couple of markers here on the cell surface. What we came to this conclusion. That was before single-cell RNA sequencing. And of course, you challenge that. If you have a higher resolution, higher throughput technology, you can challenge the old world, particularly because in this field there was a lot of discrepancy between groups and between individuals about this nomenclature. And so we and others basically worked on this, and apparently almost at the same time, we came up with 2 different new maps. You see here, this one map is basically which we now call map 1. There were like new names, and there's PDCs, you see them still, CDC1s. Suddenly there are 2 CDC2s, but there were some new cells, and you see our new map basically included suddenly what we call precursor DCs. But if you look carefully, they're not identical. Yeah? We all used, in both studies, used single-cell RNA-seq and functional data, but we came up with different conclusions. So basically continuing what the immunologists had done before, they were basically coming up with different models. And the question, of course, is if we continue doing that, and we would like to learn more about the diseases, this might not be an ideal situation. So for us, that was motivation enough to say, like, hey, how can we actually go from here to the next level and also build into this already a method that we can do that for the future. So how to build a consensus, yeah? And I bring up the Human Cell Atlas here because we're also using data from the Human Cell Atlas, building own ones. And I think this is very important also for the lifetime project, that we have both in the future, in this arena, in this community, that we're looking at and healthy and normal, but also at the diseased states. So what we did is basically we came up with an approach where we included basically strategies from both these studies, which we now call map 1 and map 2, where we used everything that they had done on the immunological level, FACS analysis, incorporated all the markers that they have basically used in both studies, which was not very easy because we used quite different markers that might explain why there was differences. And then we combined that actually with SmartSeq2. So you really have a small protein information for every cell and a transcriptome at high level information. And I think that is something that is key, that has been basically supported very much by Ido Amit, where he couples it with MassSeq, but we decided to couple it to SmartSeq to actually have even higher level information. And then, of course, what's important is how do we integrate that? And there's 2 parts to that. Is one that we integrate protein marker and transcriptome information from both studies, and what we call back mapping. We use then our information and map it back to human blood transcriptomes, single cells, as a tissue equivalent. Yeah? We would like to know our small little piece part, the myeloid cells from the blood, how do they actually look into the tissue? And this strategy is, I think, is very, very successful. And I also think that this can be applied to any kind of tissue. Blood is just a good example in the first place because it's easier to do these kind of things. So it's a showcase how we envision to do that in the future. And then you came up with a consensus map, basically. So I walk you through. These are UMAP representations. The nice thing is now we have FACS data, so we have a lot of cells on the flow basis, on the protein. Here's a million cells. These are not single-cell RNA-seq, but this is the topology of all the markers that we included. Yeah? What you can already see here is that when we take one thing, you can see this little bubble here. When we basically exclude certain cells, which you clearly see here, here's basically all cells without the lineage. But then now we focus on the cells that actually should only be the myeloid cells. Then we're losing already some cells here, which we can basically use as an information to basically look what kind of cells we have in the single-cell RNA-seq. Now, since we have index sorting, we can then decide from which areas we actually sort. And you see that we did a lot of cells for the single-cell RNA-seq, which are the black dots here in this area, because we knew that there is probably the discrepancy coming from as well as here. And we left these ones out because we knew that they're not even in the myeloid space. And you see that we did 2,500 cells with SmartSeq2. We can actually do that much cheaper now, so that's why we can afford them. So that's not a big problem here. Still expensive, but not as expensive as it was in the old days. And then you basically do another UMAP now on the single cell level, and you get this map here. And of course, now we can start to learn what are these cells, and then go back and see what we had done in the original legacy maps. The first thing we did is on phenotyping. So we looked at basically what the phenotyping was in map 1 and in map 2. And I just would like to point you to this blob here. You see that's the same color. So this is one one label, which is the non-classical monocytes, but they appear basically in 2 different clusters, which is already telling you that this might not be the same cells. There might be a contamination of another cell. If you look at the MAP2 strategy, in the MAP2, this was actually not included. So this information comes entirely from MAP1. It might explain already one of the differences. Yeah? So we can actually use the phenotyping data basically to map back the nomenclature that is in the different ones and start to see the differences once. And for that, we don't even need single-cell RNA sequencing. The many markers that we have is already sufficient to give us a first link into the differences here. And then you, of course, can map the markers on top. And as you see, these are smaller cells. This is forward and sideward scatter. We know that they're smaller. We know that they are expressing CD45RA very highly. We also see that they're not expressing a myeloid marker, so these are not myeloid cells. Yeah, so that is a contamination of another cell. And based on the other markers, the suspicion is that this is actually an NK cell. Yeah? And the reason I can bring you back in a second, because if you basically enrich your cells for studying myeloid cells, you can actually, by a certain strategy, enrich also for some very rare NK cells, and that's probably what happened. Now you can also do the overlay now on the maps of the single-cell RNA-seq data. So we map back the information from the different studies. And you can see again that MAP1 basically brings again these non-classical monocytes at 2 different sites. While this is not even marked here, it's an undefined cell that's not existing in MAP2. So there is a difference coming. And we know now from marker analysis that these are actually NK cells. I can show you that in a second. The other thing that we know is basically that these cells are not monocytes, not myeloid cells, is that when we took from the human cell atlas peripheral blood data, which is about 255,000 cells that we used based on quality criteria, made a map of these 255,000 cells, and then used the cells that we identified in this cluster as NK cells. They all appeared here. So this is basically overlaying done by the overlaying technology by the new Seurat package. And you can basically see that when we go for predicting these cells, NK cells are predicted in this quadrant, and we actually see them here. So we know now basically by many different means, and I'm showing you only 2 here, that the cells that are in one study presented as monocytes and in the other one not found are actually NK cells by this combined strategy that I presented here. We then can also go on gene level, so you can see here the clusters. If I move into this cluster 1 here, which is this cluster, there's a classical NK cell markers in these. That's why this could have been suspicious already. If you take the previous knowledge about these cells, it's very clear that these cells are NK cells. And in the end, what basically now can be said that these clusters here are not myeloid cells. The myeloid cell space is only this one. You can see that even the most sophisticated FACS sorting strategy still has contaminations. And by this backmarking strategy with the whole tissue from the human cell atlas, we can actually identify that. So the new nomenclature that we have now is basically that there are these plasmacytoidendritic cells. There are intermediate monocytes. I don't show them here because there are in the UMAP, you can see them, but if you do a trajectory analysis, you can see that there is a stream from classical monocytes via intermediate monocytes and non-classical monocytes, which clearly reflects the biology of these cells. We have still the CDC1s, that what's also very clear is that in one map1, there was already these 2 CDC2s. We now call them DC2 and DC3 according to the nomenclature from map1, which was not discovered in map2. Yeah? So both maps had basically this, not a full picture of what we know now from this more sophisticated analysis. And there is this cluster of pre-DCs. So in essence, now we have 5 DC populations and 3 monocyte populations. I didn't go into much detail here, but the 4th monocyte population is not existing, basically. So what we envision is, and this is just an example, it can be actually applied to any kind of tissue, and I think we have to do that over and over again. So you have legacy maps, and we know that already. We just collected 300 data sets in the literature from where there's also data sets from the same tissues, and we start to learn that if you look at them carefully, they're not overlapping completely. So we're trying to do other tissues as well now to do this approach. So you can combine the information both experimentally and computationally, and then add basically new layers, and there will coming up new layers. I mean, Christoph just told us about epigenetics. We will have information in the cell, and once you have that information in the same cell, you of course will have additional information about your cell. So it's very likely that we're building new maps basically integrating that. And we should basically be ready with the computational tools to be able to do that. I think one important aspect is that we have a large consortia like the Human Cell Atlas where they have the tissues as a whole, because then we can use our much more fine granular maps from very specific cells and overlay and see which cells in these maps are really truly the ones that we are expecting. So this is what we see as a very important future step. And then connect to previous knowledge. I think we should also not toss everything. For example, the whole field of immunology knows a lot about cells. Just to start from scratch is not a good idea, because there's a lot known about these cells. And then you can do the next iteration, and I'm pretty sure we're not at the end of these. Why do I think? Well, it's very simple. You know, other areas like geography or radioastronomy, they're doing that for not decades, but centuries. And they're improving still. Yeah? This is a map from Munich, 1896. And this is Google. And if you look carefully, of course, the river is still the same. Yeah? But there's a couple of things that have changed, and they're much more accurate than they were just 100 years ago. And so they're having longer time frames. I hope we're not taking so long, but I would still be seeing that as an exercise that we have to do in our systems as well. Particularly also if the technologies, for example, get better, so we can measure better things. And actually, it took me a while last night to find this map, but anyway. What? Yeah. I guess so. So now I would like to say, okay, once we have these maps, I think then we can use them as a backbone for really understanding what happens in patients. It's very easy if you do that on the campus, but what if you have patients across Europe, for example? And there we basically set up systems and also thoughts what we have to actually do to make sure that whenever we get these samples from patients, what do we have to take care of? Yeah? So this is our vision. We have to really have a lot of standardization, which we do together with companies, but also ourselves. I think we have to expand basically to spatial 3D, that's no question. And then we have to do the computation in a very standardized fashion. Yeah? And it's very clear there's a lot of things in the future. You see the medicine itself, I didn't even put, it's here, but it's in the middle somewhere. What we have to learn in medicine is that this in the future has to have teamwork between a lot of different disciplines. So educating the medical field will be a big part of bringing this really to the clinic. So the other thing is that we have established is to find those partners first that have access to interesting cohorts or have basically set up their systems and their patient cohorts in a way that we can really analyze them. Yeah? And that's not trivial, because no computation will be very good if we're not producing good data. And I can tell you that there's a huge difference between sites. And we actually take a lot of efforts to have different sites. For example, here in the COPD and the IPF field, you see that we have 4 sites where we're trying to get the same quality of data to make sure that what we're doing in the future is really somehow relevant for the patients. And we need a lot of more money for getting there, so we should be very careful in the beginning to really have good data. Otherwise, they don't trust us in the long run. So I would like to show you one example about chronic obstructive pulmonary disease, which is done by Kevin Basel. Just a short intro to you. It's a disease that's really, in addition to Alzheimer, increasing. Yeah? All other major diseases are lucky enough are decreasing at the moment. Cancer, we're very good already. So it's, of course, a lot of people still die from that, but the numbers are actually going down, and that's a good sign, death rate-wise. Not morbidity, but mortality is actually on the right track. This disease is unfortunately going the other way, and it's probably similarly heterogeneous. We have not so much ideas about what's happening. We know exactly what happens in the clinic. There's no specific biomarkers and there's no medicine. So that's, for us, was a motivation to say we have to learn more about this. If you then read the literature, it's very high-level information that we have. Yeah? It's not even clear. And, the last studies that were done for a long time, what kind of cells are there, were like 10 years old. So it's a good starting point for doing this. And the other thing that we learned is if you do it at different sites, you basically have to dissect your analysis at every step and then come back and optimize it. I'm not going through this, but just telling you that once you bring it to the clinic, suddenly you have completely different questions, yeah, unfortunately. Yeah? And if something breaks down at the early step, the rest is useless. Yeah? So that's where we're focusing on many aspects here, technologically wise. To bring it to places where there is no knowledge in single-cell technology, we wanted to have something that is very easy to transport. So was very happy when Alex Schalik from Boston basically brought this up to me even before it was published, and we were investing into the same technology. So I think we're the only lab at the moment in Europe who's also doing SegWell and have basically built up a machinery so that we can really do these kind of things for the clinical studies. So we're making these arrays ourselves, and they can be transported, can be shipped to any site. This is all what you need at the site. Yeah? You need a little training, that's for sure. And also producing that stuff is not trivial. Yeah? So we need some people that are dedicated to do that, but we have them now. And so now we can basically mass produce them and then basically apply them to clinical questions. Alex is doing the same. He has trials all over the world, even in Africa and Asia and everywhere. Does this thing work? And I think, yes, it works. So we did a comparison with 10x. We did, and here the real thing, basically, we took a bronchoscopy. This is the example of the COPD patient. We have also always controls. We enriched for immune cells because that was the study design at that time, and then did from the same sample either 10x or SegWell. If you then look individually, you find basically the major populations of cells that we are expecting. We have also always flow cytometry, so we can really control on a different method as well. And then we did the anchoring that is now provided in the new tool. And what you can see is that so far, that's the experience we have, is that we're getting more cells and different cells out in the 10x. Particularly, and that's interesting, cells that are very fragile, like neutrophils and some of the other macrophages. We see them in the SegWell, but not in the 10x, and we're still working on that and to find out whether this is always the case or not. We know from some other colleagues in the field that they're observing some similar things. SegWell is very gentle to the cells, so that might be an issue in some of the droplet-based methods, but we're investigating more into that. This is when you do clinical work. So unfortunately, I'm pretty sure Barbara has also a lot of organoids, and it's not showing everything. But in the clinic, it's sometimes really a lot of work. You see, we have done 183 patients, 119 COPDs, and then a lot of the samples drop very early because in this field there have been already people that have suggested what kind of material you should use for clinical science. And we have the quality rate, which is a recovery rate in % of what you're pushing in, blood contamination and high mucus, and you see that a lot of the samples drop out. Later on, we will use them, but really to establish ground truth, we have to basically use the best samples. We do all of them multicolor, and then we do bulk RNA sequencing, we do lipidomics and other functional tests, and some of them we can actually do to single-cell RNA sequencing already. So at the moment, we have from COPD patients about 39,000 cells, from controls, we have about 27,000 cells in the BLL. And what I didn't mention so far is we're always taking blood at the same time. So you have two organs. And also it's interesting for the clinicians, of course, to see whether blood is already a surrogate for what we see. I can tell you already one cell type, which is neutrophils. There's already changes in the blood that we also see in the BRL. And of course, the blood draw is much easier than just lavage. This is just the phenotyping data, again, multicolor flow cytometry. Just summarizing, we find, again, all the kind of cells that we're expecting. And what we saw is that in the COPD patients, you have more neutrophils. across board. We're also focusing on very early states of the disease, so we really identify the inflammatory response, and then you see that there's a very rare cell type, which are ILCs. These are these innate lymphoid cells, and the type 1 is also increased, like we can see already. We use that information to understand whether we really capture all the cells in single-cell RNA-seq. Yeah? And very often, this is not equal for each patient. So you're losing still information with all these technologies, and we at least know how much information we're losing. This is now just summarizing what we're standing in. This is also a big collaboration with Fabian and other groups. And I give you just the first pitch of what we know so far. This is the 63K cells from BAL. Yeah? And again, if you take them all together, we had no clue what kind of cells they are. So all the issues of what kind of cells are these now? So the classification of the cells. And here we also teamed up with a group or a company in Bonn. They're actually coming from a completely different field, but have now also gone into life science, and they helped us to come up with an idea where they basically we take every sample, COPD patient and control samples individual, take the raw data, have a certain filtering, which I cannot elude entirely at the moment, because this is also ongoing research. So the filtering steps are basically optimized and further optimized. We have a processed data set, and in this processed data sets, we have features, we have molecules, genes, mitochondrial genes, and there's a couple of other features that can be put in here. So we're trying to not lose a lot of information. Just by the way that if you do alignments with general pipelines, you're losing a lot of this information. We're having an annotation file that increases for information that might help us to distinguish cells. So this is the feature set, and of course this feature set could be further extended once we have other data, ATAC data or something like that, that would be available for the cell. And then we do at the same time Lavaux clustering, and then basically use things that you know already. Most of you probably, there's single R, which gives you basically identifiers based on prior knowledge. And then there's a new one that we developed, which is basically a continuation of what has been published as a package in CyberSort, but basically brought towards the single cell field. We're giving them all labels, targets, having the cell type labels, and then basically using gradient boosted decision trees with a threefold cross validation with all of these data sets to basically give a trained classifier that we then use to give a label to all the cells. So it's a classifier approach from the data itself. And since we have information, marker gene information, it's pretty amazing how good this classifier actually works on such a clinical sample. So now we know basically the cell types, and as expected, most of the cells are these red ones here, which are the macrophages. Yeah, and of course, this appears now as a large group of cells that look similar, but we know already from just some marker genes that we're looking at that this is a very heterogeneous pool, and the work that we're now doing is basically to identify which kind of functionality is in these alveolar macrophages, which is the largest population of cells. You see that, for example, this gene is all over the place, whereas you have a cell type macrophages that are highly positive for MHC class II molecules, and only some cells are positive for CXCL5. Now, the other thing that we learned is, what we didn't know is that, and I'm coming to the end, is that we find cells, and this is just from one patient, that are proliferating. And we also find cells that have clearly a monocyte-derived signature, meaning that the replenishment of these cells comes from two sources. And that was just a debate in the field. Are there only inflammatory cells moving into the disease state, or is there also self-renewal from the tissue? And we see actually both in there. And of course, now we can address that also in functional assays. And just to basically sum up, we have some indications that there is antigen processing differences, migrations. Migration is changed in these cells, which we also can test directly in lavage. And we have indications that the lipidomics is wrong, so they're also not doing their homeostatic job anymore. Clearance of the mucus is not happening anymore in these cells. So this is, I'm coming to the end, what we know now already from early stage analysis of these single cell data, that we have extravasation of monocytes into the space, we have proliferation of these cells, We have impaired cell migration of the cells itself because these cells normally move around to basically take care of bacteria that are coming in or any kind of debris. And we also know that there's this inflammatory component with loss of antigen presentation. And this brings me to the end. Basically, this is what I think is very important. In addition to the basic science that will happen in this large project lifetime, that we have this link to health and that we're setting up these kind of settings directly in patients, because we know from using cell line work and more easy systems towards these patient samples, unfortunately, you saw that we're losing a lot of material because it's not as trivial as we thought in the first place. And with that, I would like to thank my group, also the people from the platform, single-cell platform for epigenomics and genomics. I have an own group that works on myeloid cells and diseases. And then I would like to thank Andreas, who has helped a lot with the multicolor flow cytometry in these studies, and Mark Bayer, who is a long-term collaborator in the single-cell field. And I thank you for your attention.
Thank you for this nice presentation. Questions? Well, I can start with actually looking through the setup, especially like slides towards the end. I'm wondering, in your opinion, so you're using these, especially SQL at sort of 100 patient scale, right? Essentially to do exploratory studies. And do you see this as a sort of main application, or is there something kind of beyond that would require larger scales?
Yeah, so it's very clear that the first studies are proof of principle to show that we see differences and that this is meaningful to go forward. What we see already now is two ways. One is really that there are some aspects where we think that could be done, there could be easier assays to follow up on biomarker studies on larger patient cohorts. Yeah. Because you can clearly see there's a marker in a certain cell type, and you could do it by immunohistochemistry even, or something that is standard. Now, that's one pathway. The other one, what we see is, and that's particularly interesting with industry, they want to do then larger studies still on single cell because they still don't want to lose any of the information. So, and that, for me, seems a little bit like a cost issue. Yeah, but as long as we can say that we gain more information, cost should not be the factor that should stop us. Because if really single cell is diagnostically the right way to go, then we will find technologically ways to make it more simple and cheaper. Yeah, so I'm convinced that this is the case. Yeah. So, but I'm open still, and it depends on the disease. I'm pretty sure that in some of the diseases we will find easier assays for biomarker studies long term, but in others where it's complicated, you might want to stay with these kind of analyses.
I think it's very interesting to look at the plate-based assay versus the droplets, that you compare the 10x versus Sequa. And you had the statement that was sort of very weakly formulated that you seem to be able to find more fragile cells, right? If that was true, and I think it would be really worthwhile following this up, and maybe also comparing with Fox, what would we need to really then compare 10x-based studies that are so omnipresent on later on? Very often we see effects that are differential, not in terms of sort of a new population popping up, but sort of population effects. Those, of course, if we stay on the same platform, that's totally fine. But if we say then there's like an absolute differential change that might then be masked by this sort of fragility or sort of experimental.
So I think in
correct, or
in individual studies, I would not change platform. Yeah. And I'm not, that's why I'm saying like, I'm still careful because we want to analyze more samples. It could be that, this particular sample behaved like that, or the couple ones that we have. But then it's really true what we have to basically do, and this is our approach, and also in the first study, is there is some ground truth. We should not think that we don't know what kind of cells are there, particularly if it's about the immune cells. The immunologists have found out quite well already what kind of cells are there. The states might be not clear yet. Yeah? That was not possible by the markers. But the bigger populations, we're not discovering really a lot of new things. Yeah? That's also not the point about single cell. It's not about the functionality. And so if you combine any of these new technology with something where you know what you will expect, multicolor flow is really, really powerful, then you cannot run into these issues. Because if you then see that you have a lot of neutrophils, for example, which you really show by FACS, and you're not seeing them in your single cell, then you know you have to deal with it. So it's very simple to take care of these things. What's not happening at the moment, if you look at the studies, there's not a single one in this area where there is multicolor flux reported. Yeah, but easy ways to deal with it. Yeah, yeah, and we should basically also show that to the community that this is important.
More questions?